Today we're going to be looking at the metabolic health benefits of exercise. Exercise can target adipose tissue. Adipose tissue is loose connective tissue in which the adipocytes take up much of the space in the white adipose cells. They can function in insulation, absorbing shock, padding, as well as filling space around structures to support them. Adipocytes are metabolically active cells in which lipids are constantly broken down and replaced. When nutrients are scarce, lipids are broken down and the fatty acids are released, deflating the adipocytes. When nutrients are plentiful, the opposite occurs, and the fat is redeposited in these adipocytes, inflating them. When there are excess levels of lipids in the blood, and they need more storage space, mesenchymal cells can differentiate and give rise to adipocytes too. Liposuction is the removal of these fat cells, and due to the regeneration that can occur through mesenchymal cells, liposuction isn't always successful. Two different types of adipose tissue: white adipose tissue and brown adipose tissue. White adipose tissue is most abundant in the adult body and is pale and yellow in color. It serves as a storage depot for excess lipids and has many different locations around the body in which these depots are located. Based on their location, they can have different functions. Today we're going to be looking at inguinal white adipose tissue, which is located in the dorsolateral region to the gluteal region, or also known as the groin area. It plays an important role in metabolic homeostasis. Brown adipose tissue, on the other hand, is highly vascularized and has many mitochondria in the adipocytes, giving it a deep brown color, hence its name. It plays a role in thermogenesis and dissipates the energy as heat. Exercise can have many metabolic effects. It can cause the breakdown of lipids in adipocytes, causing them to shrink, reducing body mass. It can also improve glucose homeostasis and increase mitochondrial biogenesis. Previous research has shown that exercise causes white adipose tissue to show brown adipose tissue-like phenotypes, in which there is an increase in mRNA and protein levels of UCP1 and PGC1 alpha in this white adipose tissue. These factors which are involved in thermogenesis, UCP1 is a transmembrane protein in the mitochondria of brown adipose tissue. It decreases the proton gradient by acting as a channel for H+ ions, which then flow down their concentration gradient. This uncouples oxidative phosphorylation. The energy produced by the flowing of the H+ ions through the channels is then generated as heat. PGC1 alpha, which is the peroxisome proliferator activator receptor coactivator, plays many roles in the cell. It can stimulate mitochondrial biogenesis as well as enhance the expression of UP1. In the research done today, they wanted to examine the role that inguinal white adipose tissue plays in terms of the metabolic effects of exercise. What they did was they removed the inguinal white adipose tissue from certain mice through lipectomy and compared them to the control that didn't have the surgery. They then further divided these groups into groups that either had access to exercise through voluntary wheel running or had a sedentary lifestyle. They hypothesized that the removal of this white adipose tissue would lower the metabolic health benefits of exercise. They found that mice who had exercised, regardless of whether they were lipectomized or not, showed similar results. They had reductions in body mass, had increased food intake, and showed improved levels of glucose homeostasis. They also had higher levels of the thermogenesis factors UCP1 and PGC1 alpha. They also showed increased levels of mitochondrial biogenesis. We can see the metabolic effects in Figure 3 in terms of food intake and body weight. In Panel C, we see food intake measured in grams per day and with 4 different treatment groups, black being the mice that were not induced to lipectomy, whereas white representing the mice that were. SCD means sedentary mice, whereas BWR shows the mice that had exercised, and we can see that the exercised mice show increased levels of food intake compared to the sedentary. In Panel D, we're looking at body mass of these 4 different treatment groups, in which the circles represent mice that had exercised and the squares represent the sedentary mice. We see body mass weighed at the start of the experiment in initial, as well as the end of the experiment in final. And we can see that the body mass in grams was equal for all different treatment groups at the initial stages, whereas at the final, the exercised mice showed reduced body mass compared to the sedentary mice. Looking at glucose homeostasis, they subjected these mice to a glucose tolerance test in which they injected the mice with glucose to see how they reacted. On the y-axis, we can see the blood glucose levels in millimoles per liter, and on the x-axis, we see the time after the injection. We can see that at the time of the injection, all the treatment groups had the same level of blood glucose. We can see the circles represent the mice that exercised, while the squares represent the sedentary mice. And we can see at 15 minutes post-injection, the sedentary mice had higher levels of blood glucose than did the mice that had exercised. Although the glucose level decreased, the sedentary mice have continuously increased levels of blood glucose compared to the exercise mice. They then looked at two proteins involved in glucose metabolism as well as uptake, GLUT4, which is a glucose transporter, which is involved in the uptake of glucose into cells, as well as hexokinase 2, which converts glucose to G6P, which is the first step of glucose metabolism. Again, we're looking at sedentary versus exercise mice, and we can see that the mice that had exercised show increased levels of both these proteins. Overall, this shows that the mice that had exercised had better levels of glucose metabolism as well as intake compared to the sedentary mice. So overall, they saw that regardless of whether the mice had undergone lipectomy or not, if the mice had exercised, it showed similar characteristics. However, there was a difference between the sedentary as well as the exercised mice. So overall, this shows that the inguinal white adipose tissue plays a role but isn't needed for the metabolic health benefits of exercise to occur in the body. So we know that there are two different types of adipose tissue and their specific characteristics of each. Knowing this, which do you think is prevalent or excess in obesity? Also, understanding that white adipose tissue shows brown adipose tissue-like phenotypes due to exercise, which one do you think could help prevent obesity? Knowing the functions of adipose tissue, which is what is targeted by exercise, and that the thermogenesis factors UCP1 and PGC1 alpha are involved in heat production, why do you think there's an increase in these factor levels in mice that have exercised?